Kate Scow here, and this is another lecture in Soil Science 111, Soil Microbiology, and we're going to talk about the nitrogen cycle, and you will not be disappointed. First, I want to point out that this book, which I think is turning out backwards, is the ultimate book on fermentation. It's a book kind of about the history of fermented products, focusing on food, and it's by Sandor Katz. And if you love fermentation, this is your Bible. Anyway, enough advertisement. So now the nitrogen cycle, and let's try to move briskly through this. I sometimes think that my lectures end up being a bit long, but starting with what we're going to look at in this two-part lecture. This is part one, and we're going to look at a number of processes in the nitrogen cycle that is shown here. The nitrogen cycle consists of a number of microbial reactions. All of them are driven by microbes, actually, and a number of them are oxidation reduction reactions, and you already know a lot about them because they are involved in anaerobic respiration. They're involved in lithotrophy. This is actually going to be a review for you, and I think that will comfort you greatly as you see things repeated and sort of piece together some of these reactions that you looked at from a different perspective, looking at microbial lifestyles and then seeing how they fit into these larger biogeochemical cycles that have huge impacts on our planet. And particularly the way that we mess with them, then they even become more impactful. So we're going to start out looking at mineralization immobilization as the first step in the nitrogen cycle, and then talk about nitrification, then nitrate reduction, that denitrification kind of dominates. And then we're going to talk about some of the environmental concerns of the nitrogen cycle, because as I already alluded to, there are a number of them, and a lot because we as humans have really altered the cycle. And then nitrogen fixation is going to be in a second part of the lecture and is not going to be included in this one. So first, let's look at this map of total soil nitrogen. Keldal is a method to measure total nitrogen in soil, and you can see as you move west what happens in the nitrogen concentration in soil. You are right, it declines. So especially as we get down into the southwestern part of the United States, you can see big decreases in nitrogen. And actually this pretty much is just reflecting organic matter content, because there is usually a pretty good relationship between the total nitrogen content of soil and the soil organic matter content. We'll get into that a little bit more. And the other thing I want you to look at is capacity of nitrogen to both donate and give up electrons. And that makes it so interesting, so reactive, so mercurical, so changing all the time, and so well liked by microbes. The other thing about nitrogen is, as you recall from the anabolism lecture, it is a major nutrient. Right, so after carbon, and of course, there's oxygen and hydrogen, but it is the next major nutrient that microbes have to try to get. This always happens. Okay, so next slide. Let's just look at the nitrogen cycle briefly. You all have seen this, I think, for your whole lives, perhaps, but let's take a look at where is the bulk of the nitrogen. So we have these pools of nitrogen in 10 to the 12th grams per nitrogen are the units, and they are shown in these yellowish boxes, these pools of nitrogen. So where is the bulk of the nitrogen? Bingo, atmosphere, orders of magnitude higher. And I think atmosphere is made up of about 78% nitrogen, N2, I think. So huge pool of nitrogen in the atmosphere. Now, in terrestrial cycles, there's going to be some nitrogen that's in plants, and there's going to be a good amount of nitrogen in the soil organic matter as well, but nothing like what we see in the atmosphere. And then we have all these different other processes going on that are biological, and you see the numbers here are the fluxes in 10 to the 12th grams of nitrogen per year. So you can see that there's natural biological nitrogen fixation. Actually, I don't know if that's natural and agricultural. There's industrial fixation, another biological reaction, right? Because humans are the ones making the industry. Lightning contributing a little bit of this transfer of the N2 in the atmosphere into the terrestrial cycle. And then we have stuff coming out, denitrification in particular, a microbial reaction. Remember, we talked about that in class last time, about the significance of anaerobic microsites in aggregates as places where oxygen has already been used up as the first electron acceptor of choice, and then they shift over the microbes to nitrate, which in ag fields there's going to be a lot of it, and they will denitrify. They will produce N2, but also nitrous oxide, which we have a concern about. Okay, and then there's all this stuff going on in the ocean that we don't care about at all, right? Okay, and then look at this, internal cycling. And this is really important, is how can we make our ecosystems, particularly our managed ecosystems, be tight and not leak this nitrogen that can just move around in between the plant and the microbes and the soil, this closed loop. We want to keep that tight and large and turning over rapidly, and the nitrogen cycle is like playing a major role in that. Okay, so if we look at an overview of the nitrogen cycle, and you already, I know, have been exposed to this in lab, which, by the way, I want to remind you that you have the nitrogen cycle in your lab. What better way to reinforce it than running this lab in which we have essentially set up conditions that each of these major processes in the nitrogen cycle are going to dominate in those flasks, and you're going to be able to measure them with the chemical analyses that you're doing of those flasks, looking at before and after, or looking at the treatment to the control. And so you get to firsthand experience it. So pay attention to the conditions that you've set up to essentially manifest that particular leg, if you want to call it, of the nitrogen cycle. Think about what you've done to your flask. Have you flooded it? Have you added carbon? Have you added other kinds of compounds to it? Think about that, and it'll help you understand it. And as I said, you get to witness the nitrogen cycle yourself. So here we have atmospheric nitrogen going in with fixation, and going into the form of, via plants when they die, into into soil organic matter when the plants die, then release of that organic form of nitrogen into ammonium, which is going to be the major direct product of soil organic nitrogen through the process of mineralization, or ammonification it's sometimes called, making ammonia. And then that ammonium is going to be nitrified, we'll look at that in more detail, into nitrite, nitrate, and then that's going to be used as an electron acceptor, and nitrous N2 and nitrous oxide could be produced. We also have the possibility of the nitrate and the ammonium to be assimilated into the soil organic N, which is really where the microbes are. But also they can be taken up into plants as well. All right. And here are these different oxidation states of nitrogen. We have ammonium, which are in the -3 oxidation state, nitrous N2 gas, which is in the 0 oxidation state, nitrite, which is +3, and nitrate, which is +5. So you can already see here who are the donors and who are the acceptors, right? The donors are going to be the ones that are in a more reduced form, and that's like ammonium. And the acceptors are going to be in the more oxidized forms. For example, nitrate, you can't get more oxidized than that. And then nitrite in between can be either a donor or an acceptor. And by the way, that N2 gas, it looks also like it could be a donor and acceptor, but nitrogen gas has a triple bond, and that makes it quite a challenge to be able to convert from N2 gas into other forms. And that's what nitrogen fixation is all about. And nitrogen fixation is not about energy acquisition. It's about acquiring nitrogen as a nutrient. It is not a catabolic reaction. It's an anabolic reaction. And again, we will look at that when we get there. And then from the microbe point of view, here's the same steps of the nitrogen cycle, right, where we have nitrogen fixation occurring and creating organic nitrogen, and that being converted into nitrate, converted into ammonium, and then ammonium to nitrite and nitrate, and then nitrate going to N2. And then here's all these variety of different organisms that are involved in these different processes. So there's a lot of diversity, microbial diversity, that comes in carrying out these processes. Some of these processes are super general, and it seems like all organisms do them. For example, the assimilation, the taking up of nitrate or ammonium for nitrogen as a nutrient. Pretty much everybody does that. A lot of organisms mineralize, but when we get into nitrification, it's a much more specialized process. Not everybody can do it. Same with nitrogen fixation and denitrification, kind of in between. So that's interesting, right? Is to think about how much diversity of organisms are associated with these different processes. Some of these processes have a whole lot of functional diversity, as we call it, meaning there's many, many, many players who can carry out those reactions. We don't know for sure if they can carry them out under all conditions, but there seems to be like a lot of talent that can carry out some of these general reactions, whereas others are more specialists reactions. You have to be able to have a particular function to be able to carry that out. So it's not going to be as functionally diverse, is what we call it. And we worry about that if something comes in to perturb the soil, and are the organisms involved in sets of processes that are not very functionally diverse, are they going to be more vulnerable because there's nobody to take over if they get in trouble. And by the way, there's an interactive nitrogen cycle here at this website. There's lots of places to go and get extra information about the nitrogen cycle. The nitrogen cycle is a really important cycle to know and understand, because it is such a good illustration of this huge diversity of reactions that microbes engage in. It's extremely important for agriculture and for our being able to feed ourselves as humans, and it has profound impacts on the environment. So there's no getting around it. It's a hugely important cycle. It is worth investing in it and learning about it. Okay, so if we look at the nitrogen in the soil, you will find that some portion of the nitrogen in the soil is inorganic nitrogen, little N2 gas, because there's atmosphere in the soil pores, little nitrous oxide, some ammonium and nitrate in solution. But you will see that these forms of nitrogen, particularly the ammonium and the nitrate, which is what we often think about from a plant fertility point of view, and it seems to be what we think about a lot when we think about nitrogen in soil in agriculture. It's alarming what a small portion of the nitrogen in soil is in the form of ammonium and nitrate, which is what the plants need to take up in some ratio. Look at that. Together, they make up less than 1% of all the nitrogen in the soil. So there's some in the N2, but that's not going to be available unless it gets fixed. But the majority of it is in organic nitrogen form. Look at that, 77% and a little bit in the plant. So huge amount is in these complex organic forms. And if you remember back to the anabolism lecture that I hope that you spent some time looking at, you remember the forms of organic compounds that nitrogen is found in? Yeah. So that's amino acids and nucleic acids, largely are going to be the forms. So when the plant dies, this organic matter, the nitrogen-containing organic compounds are released into the soil, and then ultimately converted into organic matter, the stable form of organic material in the soil, which is different from the plant material, and we will go into that later. So you can think of the soil then as there's a bank that is the organic form where a lot of nitrogen is stored. But if you're a plant and you want to take out that nitrogen and go cash a check essentially to get it in the form of spendable nitrogen, that spendable nitrogen is in the form of ammonium or nitrate, then there has to be some mechanism to convert, to take out that banked form of nitrogen in the organic nitrogen form and turn it into this mineral nitrogen here. So that conversion of the organic form into the inorganic form is called mineralization or ammonification, like we said before. And the reverse of that, which is the conversion of the inorganic into the organic form via assimilation of that element into biomass, into microbial biomass, that's called immobilization or assimilation. So you can look at them as the flip side, right? It's going to mineralize if it's organic to inorganic, immobilization if it's inorganic to organic. And by the way, if we look at the pools of nitrogen that are making up the total organic nitrogen in the soil, you can see that we have a lot in the form of proteins, but we have a lot of it that we can't identify. It's in these pools that are defined not by their chemistry, but kind of how they behave when you extract it or how readily available they are. So my main point there is there's a lot of mystery, a lot of uncertainty about what exactly this nitrogen is looking like that's stored in the soil organic matter. Okay, oh, and by the way, I want to say that this mineralization is the conversion of either the soil organic matter form of nitrogen or plant residues, as plant residues or dead voles or whatever we're looking at going into the soil that contains organic nitrogen, as that is converted into inorganic form. And that's going to be the material that's going to be most readily available to plants. Oops. Okay, so let's look at that play between mineralization and immobilization, the organic nitrogen going to mineral. Remember, the mineral is a much smaller portion than the organic. So this is completely controlled by microbes, this ratio of how much mineralization to immobilization is going on. So they're usually going to occur together, but there's going to be a net, either mineralization or immobilization, that will depend not only on the nitrogen, but about how much carbon is also there, available to microbes when this organic input, this plant residue or whatever, humus, compost, straw, whatever it is that's going into the soil, when it's added, whether or not there's going to be net mineralization, meaning nitrogen, enough nitrogen release that plants get some, is going to depend on how much carbon is also going in with that nitrogen in that particular form of organic input. So we look at the carbon to nitrogen ratio. And we have this rule of thumb: if the carbon nitrogen ratio of that organic input that you're putting into soil is less than 25, then there's going to be net mineralization. That means there's going to be ammonium and nitrate that's released to the soil. Well, we're looking mainly at ammonium here because nitrate is another process. But if that carbon to nitrogen ratio is greater than 25, then there's going to be very little of this mineral N release, and it's going to go in the other direction and be immobilized. And that's because microbes are being fed this organic amendment, and if there is the right proportion of them, of the nutrients in it that the microbes need, then they're going to grow on it. So that would be like a good perfect ratio of carbon to nitrogen, which actually corresponds to the C to N being less than, more than, at about 25. Then they will take all the nitrogen up. Okay. That's if, all right, let's back up. I'm sorry, I'm confusing myself as I talk here. So look at it as, and I'm using my metaphors again to try to think about this or to try to describe it. So the microbes are handed a platter, and if there's a perfect ratio of carbon and nitrogen for them, then they will just use it, and there'll be no impact on the system. They'll just use everything up in that platter. But if they're given way too much nitrogen to the carbon there, then they're going to take just the right amount of nitrogen that they need to match that carbon and build up cells. And there's going to be a lot of extra nitrogen left over. And that's what they're going to release. And that's going to be net mineralization. But if on that platter there's a whole lot of carbon but not enough nitrogen to match the carbon and match the right needs that they have to make cells, then they're going to look off that platter that you've handed them. They're going to not be satisfied with just what you gave them on the platter of food. And they're going to actually look around them and scavenge up any nitrogen they can find in the soil that isn't associated with that plant material. But the plant material is going to trigger it because it's giving them carbon that they'd like to grow on. And that's when you get net immobilization. That means they will go and take away, not use up not only the nitrogen that's in the organic matter that you added, but they're going to like suck up all the available nitrogen around in the soil, not associated with that plant material. And that's going to be tough on plants because microbes are usually going to be more competitive than plants. Oops. And they will deprive them. And plants can go through these periods where they are going to be nitrogen limited, and sometimes you see them turn yellow, or sometimes they die when you're just starting to get them established. And here's some carbon to nitrogen ratios of different kinds of material that might be put into soil as an amendment from these low carbon to nitrogen ratio materials like manure and these leguminous residues with these C to N ratios that are lower than the 25. And those are going to be really good for plants, right? Because there's going to be good net mineralization. There will be nitrogen released to the system. And indeed, this is what organic farmers use, because organic farmers don't use mineral fertilizer as a source of fertility. They put in organic forms of compounds as a source of fertility, and they count on microbes to mineralize that organic form that the plants can't use into the mineral form the plants can use. So these are really good organic fertilizers. As you go down the list, so you start to see green rye, cane, I guess it's sugar cane, trash, corn stover, like the stalks of corn, straw. You can see the carbon to nitrogen ratios are getting higher and higher. And actually, those are not going to be good substrates to use as sources of nitrogen for plants, because you put those in the soil, the microbes aren't going to get enough nitrogen out of them, so they're going to go and scavenge up all the mineral N that they can find in the soil, therefore depleting nitrogen for plants around them. So you would not use these as forms of organic fertilizer. And look at sawdust. Sawdust has this ridiculously high carbon to nitrogen ratio, and it's a big immobilizer. I do want to say, though, that this is a rule of thumb. Carbon to nitrogen ratio is an oversimplification because it's assuming that there really is no diversity in the forms of organic compounds that are present. We're assuming they're all degradable at the same rate, and that they are similar in how much carbon is assimilated or whatever. But it's not a bad rule of thumb to explain this mineralization and mobilization interaction. Okay, the next stage in the nitrogen cycle. See, we've just looked at this soil organic matter going to ammonium with mineralization. We talked about immobilization, but now we're going to look at the next stage, nitrification, which is ammonium going to nitrite, going to nitrate along here, where you have a really big change in the oxidation state of the nitrogen from -3 to +3 to +5, and so that smells like what? It smells like an oxidation reduction reaction, and indeed that's what's going on. This is a very reduced form of nitrogen. This is a good electron donor, and this is an intermediate reduced, right? Because it can become more oxidized, and it's also an electron donor. And this is a strictly aerobic process, sort of, although I'm going to tell you about what rascals microbes are and how many new processes they keep coming up with. But we will just touch upon that. The nitrogen cycle is, it just keeps getting more complicated. But I'm still teaching it at sort of a more basic level that is that we know these sorts of processes are going on. We'll just touch upon some of the new developments. But usually it's an aerobic process, meaning that oxygen is used as an electron acceptor, and so nitrification involves these two reactions. There's and all of it is lithotrophic nitrification. Not all of it, but the majority of nitrification in the soil is lithoautotrophic, meaning that they're getting their energy from this inorganic compounds. First step being ammonium oxidation. So both bacteria and archaea can obtain energy from this reaction. Look at their autotrophs. Look at that, because they don't have a substrate that they can get carbon out of using ammonium and nitrate, or nitrite and oxygen. So they have to fix CO2 as their main carbon source. So they are autotrophs. And by the way, that's expensive. So they have to pay to reduce that carbon into forms that then they can use in their central metabolic pathways. So the reaction looks like this. Ammonium with 1.5 oxygens goes to nitrite. Nitrite. We're not all the way there yet. And notice that there's some protons being released, and this is a acid-generating reaction. Nitric acid is going to be produced. And then we've got not a huge amount of energy being provided by it, but it isn't too horrible. And by the way, this is a reaction. We talked about how some microbial reactions can be stopped by adding chemicals to inhibit them, and that's a good way we can study them, because we kind of shut them down as they're going along and have them dead end with the production of an intermediate compound that we can measure really well. And so acetylene block is used sometimes to study this. Now, in the olden days, we thought that only bacteria could nitrify, but there was this astonishing discovery, I think it's about 12 years ago now, that actually archaea, certain groups of archaea, can also conduct nitrification, can use ammonium as an electron donor. And not only that, they're abundant in soil. So it's not difficult to find the functional gene of ammonium oxidation in archaea in soil. but we know almost nothing about them. Very few have been cultured. Those that have are not from soil systems. They're from marine systems, or I think fish aquariums. And they're not necessarily the same organisms that are in soil. So we don't know much about the physiology and ecology of archaea, where we know a lot more about the physiology and archaea of ammonium oxidizing bacteria. But by the way, I want to also say there's an interesting story there. We used to think that the predominant bacteria that oxidized ammonium were a group of organisms called Nitrosomonas. And the reason we thought that was because they were really easy to isolate from soil. So they were the ones that would grow up on plates. So we set about studying them big time. But when we started sequencing organisms in soil and trying to understand who was engaged in these reactions, it turns out that Nitrosomonas are really uncommon in soil. They're much more common in aquatic environments or in wastewater treatment. And there are other genera of bacteria that seem to be much more abundant in soil. So we have all this information about an organism that isn't even very common in soil. And again, it's another example of how the pitfalls of culturing organisms and then basing what you're concluding about your systems on those that just happen to come up on plates. Optimum pH is neutral for these organisms, and they're pretty sensitive to lower pH, even though they may produce acidity. That doesn't mean they're good at handling lower and lower pH. The second step is nitrite oxidation, where it's an entirely different group of lithotrophic aerobic bacteria, and I don't think we know any archaea who can do this, that also use CO2 as a carbon source, but some of them can use organic carbon, actually. They're called mixotrophs. They get much less energy from the reaction, 18 kilocals instead, and we can use chlorate as an inhibitor of that particular reaction. Okay, I didn't give you I've taken away the equation, but anyway, those are the two steps. So what are the factors that affect nitrification? Well, you need oxygen for the most part, and you need a source of ammonium. Clay in the soil will affect availability of ammonium. I think we've talked a lot about availability before, and ammonium is a cation and can be held on the cation exchange capacity of soil, and particularly clay will play a big role in our agricultural soils in California. So pH, we already talked about it being sensitive to low pH, and moisture is big time going to affect nitrification because of its role in oxygen availability, and so that will be a feedback. Usually we don't observe nitrite, the intermediate, the product of the first reaction in the two-step nitrification reaction, and I think you may see that in your lab. Please, let's remember to talk about it in class and see if anybody did observe nitrite. Usually this second reaction is much, much more rapid than the first reaction, perhaps because less energy is made off of it, and so the organisms are jumping on it when it appears. And as I said, if you have a system that you use a lot of ammonium fertilizer, and so there's a lot of sources of substrate for nitrification, you're going to get big reductions in pH, making nitric acid, which is a pretty strong acid, not like sulfuric, but remember we talked about sulfuric acid when you drain flooded previously flooded, like estuary areas that the Dutch have converted into soils for agriculture, and there's that period of sulfur, H2S oxidation producing a lot of sulfuric acid. So this is kind of analogous to this, that the ammonium being oxidized also produces acid, not quite as strong as sulfuric, but it's nitric acid, which dissociates and lowers the pH. Um, Sometimes you can have like immediately after fertilizing with certain forms of fertilizer, you may actually get an increase in soil pH. You'll kind of get a roller coaster because they're basic fertilizers, but then you'll see this drop in the pH over time. Okay, and organisms involved. Remember, we talked about the Nitrosomonas was the one that's been best studied, but we actually don't find it that much in soil. This is the organisms involved in ammonium oxidation. Here's the ones involved in nitrite oxidation. So the more common ones are Nitrospira and Nitrosilobus. And then the nitrite oxidizers, or Nitrobacter is the one that does still seem to be important in soil and has been very well studied as well. But there's also some of these organisms, but they are not the same species that do nitrite oxidation as ammonium oxidation. And the archaea, they're the Crenarchaeota, and you can look up at this website more about them. It's a very exciting discovery to find that they seem to be so abundant, and people are trying to figure out under what conditions are archaea more competitive or selected for over bacteria in nitrification processes. So lots of research going on about that, because archaea are really different from bacteria. What does it mean that there's a lot of archaeal nitrifiers in soil? Okay, now also there's such a thing as organoheterotrophic nitrification, and that's a non-lithotrophic reaction where you might have amine groups, reduced nitrogen groups hanging off of organic molecules which are oxidized to nitrate. And sometimes it can be conversion of ammonium itself to nitrate. But the organisms that do this do not seem to get any energy from the reaction. It almost seems like it's a side reaction and maybe an accidental reaction, that those enzymes are just being accidentally working on these substrates that the organism isn't set up to get energy from. And the rates of organoheterotrophic nitrification are much, much slower. And here are some rates down below of an Arthrobacter, which is an organoheterotroph that can nitrify, produces nitrate, compared to Nitrosomonas and Nitrobacter, which are the organisms that will produce, involved in the two steps in nitrification. And you can see these products, and whatever you want to look at, the products that are accumulating, the micrograms of nitrogen per mil are orders and orders of magnitude higher than what the organoheterotroph does. And these are not, I'm not trying to like trick you here. These are not uncommon or atypical types of numbers in these two groups of organisms. So lithoautotrophs, right, need a lot of substrate to get energy, enough energy to make a living. So we remember we talked about the iron oxidizers. They're really down low on the tower. The drop that delta E is very, very small, so they need to turn over a lot of iron, and it has a big impact on the environment. It's the same with these lithotrophs. It's not as bad as the iron situation, but they also need to go through a lot of product to make enough energy to make a living. So it has big environmental impacts. Okay, the next step in the nitrogen cycle, so we've looked at mineralization, mobilization, and then we looked at nitrification, and now we're looking at nitrate reduction, which in this figure is shown as denitrification, which is only one form of nitrate reduction. So let's step back and look at that. So this isn't representative of all that's going on in the soil, but we have these two major kinds of nitrate reductions: assimilatory and dissimilatory. Assimilatory is an anabolic reaction, so it's involved in the nitrogen being a source of nitrogen. Dissimilatory reactions are catabolic reactions that are involved in energy acquisition. So there, in that case, that nitrate, right, which is what we're following here in the next stage of the nitrogen cycle, is going to be used as an electron acceptor. It's going to be involved in an oxidation reduction reaction in which energy is going to be made. So we can compare them in this table here. Assimilatory, the final product is going to be ammonia, which is going to be, or ammonium, which is going to be going into amino acids and also nucleic acids. So this is a reaction to, again, we said an anabolic reaction. So is it going to be sensitive to oxygen? No, because it's not being used as an electron acceptor. It's being taken up as to go into making cell material. Is it going to be sensitive to ammonium? Well, yeah, because if the organism already has ammonium, why would it want to convert that nitrate, right? Because we're talking about nitrate reduction. Why would it convert it to ammonium, and which is expensive, right? It's going to take ATP to reduce it. Why would it do that if it already has ammonium? So if you have ammonium around, you won't have this assimilatory reaction. No energy is obtained from it, and I don't know if every organism can carry out this reaction, but tons of organisms can. If nitrate is the source of nitrogen that they have, they are going to reduce it and use it. In contrast, the dissimilatory reactions, there's two kinds. We only used to know about one kind, denitrification. But now there's a second kind called DNRA, dissimilatory nitrate reduction activity. We'll talk about that a little bit later in this lecture. But let's just look at the final products of these two kinds of dissimilatory reactions where they're being used for energy, right? So they're being used to obtain energy. So there's two kinds. One, the final product is nitrogen gas, right? So you can see up here, possibly making nitrous oxide along the way. Is it sensitive to oxygen? Yes, indeed, because if oxygen is around, it will be used as the electron acceptor. If oxygen is not there, then nitrate is going to be the next electron acceptor. Is it sensitive to ammonium? No, it couldn't care less about ammonium. Why is ammonium going to impact it? Because it's all about nitrate being used as an electron acceptor. What it's going to want as a donor is going to be a carbon source. The organisms that carry out these reactions are tend to be facultative anaerobes. So they're organisms that if they have oxygen, they will use that first and then switch over to nitrate. And there's a lot of organisms involved. I'll give you a list in a minute. DNRA is, in contrast to N2 being the final product is ammonium. Is it sensitive to oxygen? Yeah, because it's being used as an electron acceptor. Is it sensitive to ammonium? No, that isn't going to really affect it. Energy is, we don't know much about this reaction. So I'm putting sometimes, we're not sure so much about it. We think that the organisms carrying out DNRA are strict anaerobes, but again, we don't know so much about it. So we're going to focus on denitrification in this lecture, focus more on the dissimilatory reaction because it's more important in the nitrogen cycle, and you can see it's what restocks the N2 gas in the atmosphere is this denitrification reaction. So it's a major loss mechanism for nitrogen from soil. Remember when we talked about aggregates in the last lecture, right? The aggregates are these microsites where even if you have a well-aerated soil, they're going to create these microenvironments where there isn't oxygen and that nitrate is going to be used as an electron acceptor. And so you're essentially going to vent some of your nitrogen fertilizer by these processes, these microbial processes inside aggregates where these organisms are using nitrate as an electron acceptor, carrying out, remember, denitrification. Okay, so let's look at the processes of denitrification. It's a series of reactions of conversion of nitrate. Ultimately, if it goes all the way to N2, but along the way, through a series of these reactions, there's also going to be produced these intermediate products, nitrite, nitrous oxide, nitric oxide, nitrous oxide. This is, and then of course you need an electron donor to be able to carry out this reaction. So I'm just putting glucose here as an example. So this is just a big sort of descriptive equation that pulls it all together. So this figure below shows you a bacterium that can carry out denitrification, and looking at the genes that are involved in these different steps of denitrification. So remember, we're going through a series of oxidation reduction reactions. So we have nitrate being converted to nitrite, and there is what we call the NAR gene that's responsible for that. Then nitrite is going to go to nitric oxide to NO. NIR is involved in that reaction, is the enzyme involved in that reaction. Then the NOR gene is involved in the conversion to nitrous oxide, and the NOS gene is involved in the final step of the nitrous oxide to N2. So a lot of denitrifiers will have all these genes, and we'll be able to carry out the whole reaction. But there are some organisms that only go like to nitrous oxide or to NO. So not all organisms have it. So why am I showing you all this? It's not for you to memorize all this, but it's to see that, look, here's all these functional genes, right, that are responsible for these different steps in the process of denitrification. So you can go and target the genes that encode for these enzymes, right, for these different steps, and measure them in the soil. You can extract DNA from soil and then see how many, like using quantitative PCR, what's the copy number of these different enzymes, and that will give you a lot of information about the ecology of what's going on in the process of denitrification by being able to target the functional genes and not just the phylogenetic genes. And obviously, if you've got like not much of this gene in the soil, but a whole lot of these genes, then you're going to get a little bit worried, because you're going to see like, whoa, there's not much capacity for that nitrous oxide to convert to N2. So it gives you a lot of ecological information that might help you make management decisions. And remember, we don't like nitrous oxide because it's a very strong greenhouse gas. Okay, a lot of different organisms can denitrify, including some organo and lithotrophs, and these are just some names of these facultative anaerobes that can carry out these reactions. And of course, we know a lot more than I think when this table was put together. But there's a lot of diversity of organisms that can denitrify. Okay, so what regulates denitrification? What are the factors that determine like whether or not it's going to be a high rate or a low rate or no rate at all? Well, we can look at what's inside the red circle. Obviously, you need carbon, you need an electron donor, and you need nitrate, right, which is the electron acceptor. And also, we need not to have oxygen around. So what I like about this figure is then you can sort of back up and say, okay, what are the processes or the factors that are responsible for like how much oxygen is going to be in the soil? And so that's going to be very determined by water content, which is going to be determined by how much rain and the texture of the soil, and plants using up the oxygen, or plant material, plant residues going in and being a source of carbon. So anyway, here are these this cascade of all these different factors that are ultimately going to, the plants are going to use water, that determine how much oxygen is in the soil. And also organic matter decomposition, respiration will also affect how much oxygen is left over. And then the same with nitrate. We know that it comes from nitrification, ammonium, and the ammonium is coming from the organic matter. This is mineralization. That's very important. And then the water content is going to affect the availability of the nitrate and the carbon, the diffusional path lengths, and availability to microbes. And then we've got these different factors that are going to affect how much carbon is is available. So it gives you a nice kind of organizational roadmap, letting you think about what are the conditions that denitrification are going to be higher or lower in. All right, now this slide, this is a pretty fantastic study that was done that really illustrates this idea of this tremendous heterogeneity in denitrification rates in landscapes, in this case in an agricultural field. And what you see here is essentially a dissection of where in a soil column the bulk of denitrification is going on. So there's some numbers you see here. At the bottom of these little rectangles that represent these smaller and smaller increments of this one original soil profile that was collected to 15 centimeters. There you see the grams of the mass of each of these soil increments. So what we're seeing here is a big soil core that's divided up into smaller and smaller units, going from about 100 grams down to 5-gram increments, and then breaking that apart as well. And then the other number you see is at the bottom of each of these columns, which is a denitrification rate in nanograms of nitrogen per day. So you can see there's over 5,000 nanograms of nitrogen being denitrified per day in the whole soil column. Now, when they divided it up into 3 sections, equal sections, you can see that there was hardly any denitrification rate in 2 of the sections, but in the top layer, there was still quite a bit of denitrification going on. So then they broke that up into smaller increments of soil, and you can see that these rates are really small in most of them, but in that top layer of the soil, there was as much denitrification as was going on with this almost 4,400 as was going on in the larger chunk of soil. Don't worry that this number is a little bit bigger. That's just variation in data. So then they dissected that little 5-gram sample of soil and pulled out this bit of decomposing leaf. And then also looked at what was going on in the remainder of the soil in this small subsample. And you can see that around that leaf, there was huge rates of denitrification. The bulk of the denitrification that was going on in the whole 15-centimeter column was going on around that leaf, and that there was very little denitrification even going on in the soil immediately around the leaf. So this goes to show you that there was an incredible hot spot of denitrification going on right around the leaf. Why do you think that was? What did that leaf supply? I think you said the right answer, which is electron donor. It provided a source of carbon. That was what was driving this rate of denitrification. There was presumably nitrate in all the other samples of the soil, but not sufficient carbon to result in a very high rate of denitrification, because you need a donor and an acceptor, remember? So you can imagine then how difficult it is to go out in the field and measure a representative rate of denitrification in a soil that you might want to then take to policymakers and say, look, this is how much agriculture contributes to denitrification and nitrous oxide formation, therefore we should do this. When you have such heterogeneity, it's really hard to get those numbers that you can rely on that can then go into making policy. Okay, I wanted to point out another video that you can go and watch. It's really dry and it's really mainstream, but it's really, really, really just like clear. So you can go and look at that to get more information on the nitrogen cycle. I recommend it even though it's dry. Okay, and I warned you that there's a lot more going on in the nitrogen cycle than what I have talked about, and I totally don't want to go into detail on this, but do you see what a nightmare this is? We had our nice, obedient nitrogen cycle with organic nitrogen going to ammonium and then going to nitrate, and then that going to N2. Well, it turns out there's other shenanigans going on that are involving different combinations of some of these forms of nitrogen in different ways, including an aerobic denitrification pathway, nitrifiers involved in denitrification. Like what? Nitrifiers are lithotrophs. Denitrification is nitrate being used as electron acceptor. What the hell is that? But there's something going on there. There's fungi that may be involved in denitrification. And there's something called anammox, which is number 5, which is bringing ammonia and nitrite together to form N2. Again, what? And then there's also nitrous oxide production that can occur that we've been thinking is mainly during denitrification, but it can also happen during nitrification. And then there's also DNRA. Remember, we talked about that with nitrate reduction as the other dissimilatory pathway, and let's just glance at that for a minute. But meanwhile, I just want you to know that there's way more going on than what I just presented to you. How important these other pathways are in soil, we really do not know. Some of these pathways have only been observed in like wastewater treatment systems or in aquatic systems. And even if we find them in soil, we don't know how significant they are. Although I think nitrous oxide during nitrification, we do know that that's going on a lot in soil. But anyway, let's just revisit the DNRA. And sorry, there's all the little details about these guys. I forgot to do that. Okay, so DNRA, dissimilatory nitrate reductase activity. So we don't really know exactly why organisms do it. Maybe ATP is made from it. Do you remember what the byproduct of DNRA is? What the final product of it is? It's not denitrification, remember. So it's not N2. That's right, it's ammonia. It's ammonium. So maybe organisms do it to detoxify nitrite, because nitrite can be toxic to a lot of organisms, or maybe to regenerate that old NAD, dump electrons in some product that comes out. It tends to be associated with energy-rich, like carbon-rich environments that don't have really high concentrations of nitrate. And a number of organisms in pure culture have been found to carry out this reaction. A lot of them are obligate anaerobes that we know most about, although it seems maybe it's found in other organisms. There's been a little work done on soil showing the presence of this kind of reaction, but not very much work at all. And it's kind of shaky. We don't know really how important it is. But let me tell you something. If it is going on, or if we could make it go on more and we could make more of that nitrate not go to N2 gas or nitrous oxide, so not be vented to the atmosphere or not produce that greenhouse gas, but have it go back to ammonium, wouldn't that be spectacular? Then you'd have that closed loop that we looked at the beginning of the nitrogen cycle. Remember that internal cycling? If more of this was going on, there'd be much less opportunity for leaking of nitrogen outside of an ecosystem. So there's a lot of interest in it, although, as I said, the evidence for it is not that great, for soil, that is. Okay, I want to just rattle through some environmental issues of nitrogen. We as humans have really wreaked havoc with this what used to be a much better behaved cycle, and we've created all kinds of opportunities for microbes to be co-agents in this generation of environmental problems, but also they're also can be involved in some of the solutions. We have altered the nitrogen cycle big time. What we've done is taken a lot of that inert N2 gas that was just floating around in the atmosphere, 78% of the atmosphere, and we have converted it into now biologically available forms. We have brought it into the biosphere. And how have we done that? We've done that largely by increasing fertilizer production. Is one way. And so you can see this figure that shows, in the olden days, in 1920, 40, 60, 80, right? And you can see starting in the 40s and increasing significantly and continuing to increase is much greater production of synthetic nitrogen fertilizer that we'll talk more about when we talk about nitrogen fixation. Legumes and green manures have always fixed nitrogen. They continue to do it maybe at lower amounts. There's always been natural biological fixation going on. Perhaps that hasn't changed. Perhaps it's gone down as we've destroyed some of the other natural ecosystems, and lightning is also a source. But look at the other source of nitrogen into the biosphere that's under this anthropogenic. That means humans generated it, right, in the top. And that's fossil fuel combustion. So that's the other big thing we've done, is we've taken all this nitrogen that was stored, buried deep in the subsurface inside fossil fuels, and we've combusted it. We've burned it in our industrial processes and in our gasoline. Yes, you and me have done a lot of this as well, releasing what used to be buried, not engaged nitrogen now into the biosphere. So this has led to a lot of different impacts, including eutrophication of water bodies. This has been well studied in the past, of bringing in a lot of nitrogen. Sometimes it's phosphorus as well, but essentially fertilizing systems that are oligotrophic, and suddenly there's new nutrients available from this misplacement of nitrogen, and you get algal blooms that explode and then die and then are decomposed and use up all the oxygen and kill all the fish. There's also acidification due to some of these nitrogen processes generating acid that has also occurred. There's been nitrogen brought into forests, just like kind of with the lakes. These are forests are oligotrophic systems that aren't used to seeing a lot of nitrogen, and now they're being fertilized through the atmosphere, through deposition. This is also happening with our chaparral systems, like around LA and the LA basin, due to the cars releasing so much nitrogen that then falls out in these really very specialized nutrient-poor environments. Like they've figured out, those plants, how to deal with there not being a lot of fertility there, and suddenly all this nitrogen is falling out of them falling on them out of the sky, and you'd think, well, that's great, isn't it? This is just what they needed, but it isn't what those ecosystems need, because it ends up bringing lots of problems to them, like increased rates of disease because they become more vulnerable, or insect pests start to come, or you get shifts in the community to more weedy things that are really good at jumping on opportunities of nitrogen and shutting out some of the indigenous species. Also, we've had, and we've talked about this already, which is why I'm doing it last, nitrous oxide, nitric oxide, and a little bit of ammonia, particularly like from dairies, where we've released these compounds into the atmosphere because there's just a lot more of it going on. Good website here if you want to look more at this human alteration of the nitrogen cycle. And by the way, this is a really good review article. It's kind of old now, but it's a great article if you want an overview of how humans have changed the nitrogen cycle. All right. I think I'm going to skip over this, but we're talking, it's about the same, it's the same thing here, but it's showing it in a lot more detail, essentially comparing those sizes of pools of different forms of nitrogen and the rates at which these compounds are formed or consumed in 1850 compared to, in the last decade of the 20th century. So you can just look and show yourself that there's been these big changes shown by the sizes of the arrows in terms of how much of these original compounds, in the original forms and locations have moved into different places in the terrestrial biosphere. And we have huge nitrate pollution of groundwater, and this is a problem throughout the whole U.S. This is not even the most up-to-date map, but it shows you areas where there's low to high risk of nitrate contamination. Pink is high risk, which has to do with how much nitrogen fertilizer there is and the possibility of, climatically for that nitrogen to move from the soil where it's applied as a fertilizer into the groundwater. And you can see big hot spots are in the Midwest and then also California, our own Central Valley. And we're going to just touch on that because I want you to understand what's going on locally. So nitrate is very mobile. It's an anion. It's not going to soar to the cation exchange capacity. And as soon as nitrate is formed, then boom, there's opportunity for mobility. And it will, as we already talked about, can go into surface water through runoff and cause eutrophication. Also, if nitrate goes into the groundwater, it's associated with this health problem that particularly affects babies, and it's called the blue baby syndrome. And it involves the formation of it involves the creation of this disease, methemoglobinemia, which is this blue baby syndrome. And what happens is if babies are drinking water that is high in nitrate, then that nitrate is going to bind to the hemoglobin in their bloodstream instead of oxygen, right? The hemoglobin is what moves oxygen around in the body. But nitrate can also bind to it, particularly if it's in high concentrations, and that can lead to oxygen deficiency in the baby, and the baby turns blue. And this illness, this health problem, is what is used in regulatory guidelines of setting goals or targets for how much nitrate is acceptable in groundwater, is due to the potential incidence of this disease. Now, there's a lot of other things that nitrate, and other health problems that are associated with consumption of nitrate that isn't so much reflected yet in our regulations. But as time goes on, there's more and more studies that show other health problems as well of ingesting high concentrations of nitrate. Also, nitrosamines, which are carcinogens, can form from nitrate, like in water being combining with other kinds of foods or with chemicals in the environment. So how do we manage to reduce nitrate reduction? I mean, ways that we know already is to avoid excess irrigation that would move nitrate down below where the plant can take it up. Use less nitrogen, time the use of the nitrogen better. People used to fertilize a lot in the winter, and then the rains would come and move that nitrogen around before the crops were planted and taking it up. And I mean, there's been improvement in that throughout the United States. It's also possible to use slow release fertilizers, which are kind of expensive, but they're formulations that are in like resins or some kind of immobile substrate that just slowly is degraded over time, releasing nitrogen in smaller doses over time. Inhibitors, which are poisons that specifically target nitrifier organisms, that end up through a couple of different mechanisms, just shutting down their ability to nitrify, converting ammonium to nitrite. And so this is like another chemical that would be put into the soil. So it's got that issue associated with it. And also there are a lot of organisms that will use the nitrification inhibitors as carbon and energy sources. So they will, in some cases, the nitrification inhibitors don't hang around long enough to be very efficacious. In rice, this is just sort of a sidebar, fertilizer would never be in the form of nitrate in a flooded rice field. Why is that? That's right. that if it was added in the form of nitrate and there's not a lot of oxygen around because it's flooded, then the organisms in the flooded rice field are going to use nitrate as an electron acceptor, and a lot of your fertilizer is just going to go to the atmosphere. So traditionally, rice is fertilized with ammonium or urea. And in groundwater remediation, since there is a lot of contaminated groundwater, just reminding you, and we've already talked about this a lot, would you add oxygen to clean up that nitrate? No way, right? To clean up that groundwater, but this isn't done very much. But if you were to clean up that groundwater, you would want that nitrate to be used as an electron acceptor. Therefore, you need to add a donor. Any sort of organic compound that you have kicking around, so molasses or residues from, byproducts from making, processing sugar, or I don't know, there's a variety of different sources of waste, but it isn't used. This remediation of nitrate is not very commonly done. Okay, and then let's talk about the gases. So both nitrification and denitrification result in metabolic products that are greenhouse gases that destroy the ozone layer. And here we've got our mineralization going on, and ammonium going through nitrification to nitrate, and then nitrate going to N2 through denitrification, showing these leaky pipes. That's a metaphor to describe what's going on. These places where either nitrous oxide, which is the more commonly occurring intermediate that could leak to the atmosphere, but also to some degree it can also be nitric oxide, which is also volatile and can go. And you can see these things being formed in both nitrification and denitrification, which are these very potent greenhouse gases. Okay, and then I'm not going to spend a lot of detail on it, but there's a few years ago, it's 2012 now, this very monumental study was done, and this report to the California State Legislature called SWRCB SBX 21, which was to make an assessment of what is going on with nitrate contamination of California's drinking water, focusing on the Central Valley. And you can go look up this big report, which has been sort of a game changer for how we think about nitrogen. So in the past, we know there's a lot of nitrate pollution in groundwater, but everybody was pointing their fingers at each other saying, oh, it's coming from septic tanks, or oh, it's coming from fallout from automobile exhaust that then hits the soil and leaches, or yeah, we know fertilizer can contribute some to it, but there's all these other sources as well, so don't gang up on agriculture. They are just one of many, many sources. But this study, through using a variety of different measurements and also models, and just gathering all kinds of data from governmental reports, governmental monitoring, but also interviewing farmers, and just this huge, huge report. They found that the two largest sources of nitrate that leaches to the groundwater, 90% come from either fertilizer use or animal manure. Animal manure also applied to cropland, so associated with dairies. And they also said that it is possible to reduce that by improving fertilizer management and water treatment, but some of these strategies to do that would be pretty expensive. Here's the remediation aspect. Directly removing nitrate from groundwater once it's there, they concluded is very, very expensive and technically not really very feasible. So that's why I think you don't see it used very much. And then they said that where the target really needs to be is in water treatment, in treatment after the fact of these the water that's coming from these contaminated groundwaters, and finding alternative water supplies. Not that the upstream type goals need not to be achieved as well to reduce the nitrate that goes into the groundwater. But as of now, we have to focus more at the tap or in the drinking water treatment to be able to reduce that nitrate pollution and reduce exposure of people to nitrate. And there's coming out of this is a new regulatory program called the Irrigated Lands Regulatory Program. And you can go and look at it here at this Cal EPA website. And there, what is being regulated and is actually starting to be deployed is a set of actually coalitions that help span the gap that is between farmers and regulators. So groups of people that will help to interface and communicate back and forth between the two groups. And then most important is this A to R ratio, is this metric to be able to look at progress in the control of sources. And that A and R is essentially this ratio of how much nitrogen is applied to a particular field. So this would be for a farm, divided by how much nitrogen is removed during harvest. And then if it is a woody plant, how much nitrogen remains in the wood of, say, almond trees or grapes, vines, if it's that kind of crop, at least for the time being. Whereas like in tomatoes, this second number wouldn't be relevant. So it's essentially looking at inputs versus outputs. So this means that there's going to be greater monitoring and reporting back from farmers that needs to occur to be able to provide information on this AR ratio. There's going to be monitoring of groundwater nitrate concentrations over time that the state is going to support. And there's going to be money that is going to come out for research to help figure out how to keep agricultural yields up, but to decrease this A to R ratio, right? You want to get this lower so that there's less potential for leakage of nitrate. Okay, so I have a couple of discussion questions that you can think about. And we're not going to look at it right now in this lecture, but I think these are pretty interesting questions that make you think about the whole nitrogen cycle and how could you manage it to achieve these two different kinds of goals. And there's supplementary information, so just figures showing you where you might expect to see denitrification versus DNRA. Some ecological niches for these different types of nitrogen transformations that are a function of how much carbon, how much nitrogen, and how much oxygen is in the system. And then a beautiful cartoon of the nitrogen cycle, just as a treat for you to look at. And then some of the particular reactions involved with the mineralization reaction that we talked about. Just reminding you that mineralization is a conversion of organic forms of nitrogen to inorganic forms, and what are those organic forms? Are like proteins and nucleic acids and peptidoglycan actually as well, that go through these series of reactions to produce the mineral N. And assimilation pathways for nitrogen getting into the cell, that is really, really not very pleasant looking. But if you go back, this is for those of you who are nerdy and want more, you can look at this and compare it to the figure that we looked at the end of the metabolism lecture. But this is more from the impact of thinking, from more of the perspective of how nitrogen gets into the cell. And some little information about nitrogen treatment in wastewater, where you might want to alternate aerobic and anaerobic conditions to be able to maximize the removal of nitrogen. And that's it, folks.